Right. Lesson 2 of genetics and inheritance. I'm trying to keep the videos short as much as possible because for every 17 minutes they take 1.6 gigs. So, but we're gonna go now into we start obviously with our exam guidelines. Let's blow them up. Right. Remember what I always say to you. Always have your exam guidelines next to you. Right. What do they say? Mention of Mendel as the father of genetics. That's what the current guidelines require you to do. Are we together? And then for the 2015 ones, they want you to outline the experiments conducted by Mendel. Mendel is the father of genetics, by the way. We're going to go into him in more detail. But now, this part here, these are the 2015 guidelines, which are no longer relevant now. And they want you to outline the experiments conducted by Mendel. Whereas in the 2017 guidelines, the latest ones, the only thing that you have to do is to mention the mention of Mendel as the father of genetics. I don't know now how Mendel is going to be mentioned here, in what context they're going to be mentioning Mendel as the father of genetics. So, like what I always do every year, we're going to go into Mendel. May be relevant, may not be relevant. This thing is, as you can see from what's written, the mention of Mendel as the father of genetics, I'm not too sure what mention. I don't know if they want you to state that Mendel is the father of genetics. I don't know when they say what do they want you to mention about Mendel. So every year I keep it safe and simple. If Mendel is going to come in any form in the exams, I would rather we go and then we talk, not outline exactly, but talk about, let's talk about his experiments so that you get into how the basics, the foundation of genetics and what it is all to do with. So we're gonna go into Mendel for this lesson 2. We're gonna go into Mendel and then we're gonna work with Mendel. Don't get too obsessed with it. I'll even tell you where you don't have to get obsessed with it. But I feel that it is relevant that the things that I prepared for you, that we cover them in some good detail. Remember that our aim always is to get 150 out of 150. That's always our aim, and our aim shall not change. Lockdown or no lockdown, COVID or no COVID, our aim will not change. Right, Gregor Mendel, 1822 to 1884. Right, Gregor Mendel is considered to be the father of genetics. In other words, for us to know what genetics is and what it is all about, it is because of Gregor Mendel. He was actually, I think, an Austrian monk. I think, I'm not sure, but I know that he was a religious figure. Right. So what makes Mendel to be special? Mendel is the cat who performed experiments on pea plants to study the inheritance of characteristics from one generation to another. Performed experiments. Let's see if this thing is working. Work with me. It's not working, but anyway, it's fine. He performed experiments on pea plants to study the inheritance of characteristics from one generation to another. And through these experimental findings, he formulated the basic laws of genetics. Now, listen, Mendel chose pea plants for his experiments. And I want you to put it this way. You can't take a complex organism and then use the complex organism to study remember what genetics is? The study of heredity and variation. So if we are studying heredity, we cannot be looking at a complex animal with a long life cycle that has a long life cycle to the point where, for example, I'll give you an example because here I'm rambling. If we take human beings, we can't study how stuff is going to be inherited from your mom, or at least one scientist, or a team of scientists can't study how a team of scientists at any given moment and come up with conclusive results. When we take a human being as an example of a subject to be studied, to see how inheritance occurs of different characteristics, because we live far much longer on earth. So you want to have a particular subject that is going to be able to give you many generations during your study period, so that you can now then present results from the many observations that you've made, and those results will be what? Reliable. Remember reliability, increasing the sample size or repeating. So he chose pea plants, Mr. Mendel here. And Mendel's choice was very good. Why? Because it shows clear, and I put a tick there for emphasis. Remember I think I actually remember seeing this in an old past exam paper on why Mendel chose, it can be national or it can be some particular province, I don't know, but from years ago, on why Mendel chose pea plants. Why is it that they were a good choice? And the answers were because they showed clear, tick, observable, tick, and contrasting, tick, characteristics or traits. Contrasting means that they are from A to B, it is so different. From A to B, it is so different that you can actually see how different it is. Like saying A is white and then B is black, or red and green. So that's what contrasting means. So P's have very clear differences. They are, and those differences you can observe them. You will find that there are characteristics that we have that are not easily observable. You can't observe if a person is a tongue roller or non-tongue roller up until they start stick out their tongue and start doing it. Yeah, and so observable, they're very observable, pea plants you see as we go into this lesson, and then they also have contrasting traits. You know, you don't have intermediates with pea plants, which is a good thing because, and out of these pea plants, Mendel, when he looked at them, he looked at 7 of these contrasting characteristics. So what are these? You looked at pea plants that were tall and short. You looked at pea plants. On pea plants, you also looked at the smooth and the wrinkled seeds. In other words, there are 2 types of pea plants. Some will produce smooth seeds, round seeds, and some will produce wrinkled seeds. I think you've seen this if you go to, with your mom to shop. You'll find that you're going to have these situations where some of the peas that you buy, they're not rotten when they're not stale. It's just that the seeds are wrinkled because it's part of their genetic structure. And then you also looked at pea plants that produced yellow seeds and those that produced green seeds. You also looked at pea plants that gave you inflated and those that gave you constricted pods. And then you also looked at pea plants that had green pods and those with yellow pods and purple flowers and white flowers and axial and terminal flowers. Now, let's go to the images. Where are you? Grade 12. There we go. 22. Check this out. These are the 7 traits or characteristics, whichever term that you use is fine. Traits and characteristics is the same thing. So when Gregor was doing his thing, he was looking at 7 different characteristics. No, that's too small. Let's do this. So we can zoom in. Let's open this with Bingo. Now, walk with me. Remember what we just said? Mendel did what? He looked at 7 characteristics. Now, the 7 characteristics are flower position, seed color, seed shape, pod shape, pod color, and then stem length. These are the 7 characteristics. 1, 2, 3, 4, 5. Oh, and flower color. So 3, 6, 7. 1, 2, 3, 4, 5, 6, 7. Those are the type of traits that he looked at when he was studying peas in his experiment. And what was he studying for? The heredity of these traits and the variation within one generation to the next generation of the same given traits. Now, you will find that in peas, you have peas that have purple flowers and those that have white flowers. This is very clear. This trait of flower color is clear, it is observable, and there are contrasting differences between these. Because purple is purple, you can see what purple is and white is, right? And then the flower position, you either have axial flowers, those that are okay on the sides, or you have terminal flowers, those that are okay at the top. And then seed color, you have yellow seeds and green seeds being produced by different PE plants. And then the seed shape will either come as round or it will come as wrinkled. The pods will either come as inflated or as constricted. The pod color is either green or it is yellow. And the stem length is either tall or the plant is dwarf or short. So you will find that these 7 characteristics, each trait has got 2 different forms in which it can appear. Pick any trait here. If you look at seed shape, either pea plants will give you peas that have round seeds or that have wrinkled seeds, no third option. Either you have flowers that are found on an axial position or a terminal position. You have stems, or if you look at the whole pea plant as a whole, you either find that the pea is either tall or they are dwarf or short. You don't get pea plants that are going to be middle there between tall and no. It's either you are this or you are that, making it very clear, observable, and also having contrasting differences. So this is what Mendel looked at. I will send these notes to you via D6, but this is what he looked at. And these 7 characteristics, when he looked at them, he ended up coming up with observations, just like any responsible scientist is supposed to do, coming up with observations on and conclusions from the various experiments that he had done. Now, remember what we said? We said that the reason why this was a good choice, pea plants were a good choice, because they are showing us what? A clear, observable, and contrasting differences. This is why pea plants were a very good choice. But another advantage of pea plants is that naturally themselves, they self-pollinate and do not cross-pollinate. Now, why is it that, why was that important? It's because it was Mendel now. Now, think Mendel in a greenhouse. And he's got a bunch of pea plants. He's got a bunch of pea plants within the greenhouse. And then, with varying characteristics. Let's start simple. Let's start with the basics. You're either having tall plants, and then you are also having short pea plants. And this brother here has got pea plants within a greenhouse, and he's doing his experiments. Now, whatever experiments that you're doing, you don't want external factors like pollinators then coming in and messing up with your experiments. Now, because pea plants are naturally self-pollinating, this meant that he could basically be the one who takes a brush, goes to, let's say that he wants to see what happens if a tall plant and a tall plant end up, if pollination occurs between two tall plants. What did he have to do? Because pea plants are naturally self-pollinating. All he had to do was to take a brush, go to a tall plant and brush off some pollen from that plant, and then go to another plant and then brush the same brush with the pollen now onto the sticky stigma of another plant. So he always knew that whether in his absence, whether he's there or in his absence, whatever results that he was going to get, whatever offspring of peas that he was going to get, they were a result of him himself being the one who was in control and in charge of the pollination that occurred. And this is an advantage, right? This used to be a past exam paper. I think one of those years ago, I think when 2015 exam guidelines were still valid and the ones that were still there. But remember what we said, and let me stress this. Let me stress this. Remember what we said. We say that the 2017 exam guidelines are our life science Bible. Let's not go away from it. So they only require us to what? To a mention of Mendel as the father of genetics. I gave you my reasons why. I don't know how far that goes. Does it only go to stating who Mendel is, or it goes further than that? So to play it safe, I will go back to the exam, to the 2015 guidelines, and I hear that they outline experiments conducted by Mendel. That is not there in there. However, for you to get the gist of genetics, for you to understand the basics so that we can deal with what's coming, we need to take our porridge first before we go to the pub. So, here we go. What else from these experiments? So what did he do? This meant that Gregor Mendel himself could cut off the anthers containing pollen and then perform cross-pollination by himself. He could perform cross-pollination by himself using pollen of what? Of his own choice. Now, how did he do this? He painted ripe pollen onto the stigma with a small brush and could thus control the cross. And initially, Mendel did his experiments with parent plants that were true breeding for a specific characteristic. Initially, Mendel did his experiments with parent plants that were what? That were true breeding for a specific characteristic. What are we talking about here? What do we mean when we talk about the term true breeding? A plant is considered to be true breeding for a specific characteristic. For a specific characteristic when self-pollination takes place and all the offspring produced have the same characteristic as the parent. Now listen to this. Remember, let's go back to our basics. We said that you have, if we pick any one of these, if we pick any of these characteristics, let's remove this. There we go. I like using that because height is something that you can envision, like something that you can visualize. Now, walk with me. When you have pea plants are either going to come as being tall or being short. Either tall or short. Now, if you allow a tall pea plant to self-pollinate, for a tall pea plant to do itself, self-pollination, for a tall pea plant to self-pollinate, in other words, the pollen from that plant falls on the stigma of the same plant, and then self-pollination occurs. And then if that tall plant gives you offspring that are tall and short, it is not a true breeding. It is not true breeding for the characteristic of stem length. Let me repeat that. If you take a tall plant, a tall pea plant, allow it to self-pollinate, and then it produces offspring, and the offspring are, some of the offspring are tall, and then some of the offspring are short. This plant cannot be considered to be true breeding, because a true breeding plant will produce, if it self-pollinates, a true breeding plant for height will, and the height is tall, is considered to be true breeding if it is able to self-pollinate itself and it produces offspring that are all tall. That is a true breeding plant. I'm going to use an example that you should never use. And then, okay, let's put it aside. I'll wait to hear first if you have questions. And then, a short pea plant can only be considered to be true breeding if it is allowed to self-pollinate, and then it produces only offspring that are also what? Are also short. In other words, this plant that is able to produce, after self-pollination, that is able to produce pea plants that are having the same, or offspring, having the same trait as the parent, then we say that plant is true breeding. Now, listen to this. Walk with me here. Let's go back to the definition of what a pea plant is. A plant is true breeding for a specific characteristic when self-pollination takes place and all the offspring produced have the same characteristic as the parents. Now, let's do another one. Let's do another trait. If I take a plant with yellow seeds that produces yellow seeds, a pea plant that produces yellow seeds, I allow it to self-pollinate, and then it gives me yellow and green seeds. Is that a true breeding plant? No. It is not true breeding for seed color. Right. If I take another pea plant that gives me green seeds, and then I allow it to self-pollinate, and then it gives me all the offspring that it produces are all green, then we say that it is true breeding for the green seed color. Now, bear in mind something. If we go back to our original definition of what a true breeding plant is, a plant that is true breeding, this is the first question, by the way. A plant that is what? That is true breeding for a specific characteristic. Why am I putting emphasis here? Because you know that you will see that within a plant, each pea plant, can you imagine that each pea plant has got, let's take one pea plant. Let's take one pea plant. Let's take one pea plant. Let's look at one pea plant. Right. Let's move this, that. There we go. Let's say our pea plant A has got, it is white. Let's make a pea plant. Let's make a pea plant. It has white flowers. The flowers are positioned terminally. It produces yellow seed. It produces wrinkled. The seed shape is wrinkled, and then you're gonna have the pods being inflated, and the pod color being green. And this brother here, this pea plant, is what? Is tall. So if we have a pea plant here, if we put a pea plant here and then we say, describe the pea plant, we will end up saying what? We would say that this pea plant has white flowers that are positioned terminally, and it has yellow seeds, and the seed shape is actually wrinkled, and the pod shapes are inflated, and the pod color is green, and the pea plant is very tall. Now listen to this. Let's go back to what our definition is of what true breeding is. A plant is true breeding for a specific characteristic. This means that for this plant here that we've now made, this plant, that is white with terminal flowers with yellow seeds that are found within wrinkled pod shapes, seed shape, and then the pods are inflated and they're green in color, and the pea plant is tall. For this plant here, it doesn't mean that if this plant is true breeding for yellow pods, or it is true breeding for green pods, whichever. It doesn't mean that if it is true breeding for pod color. In other words, let's say that it's green. Remember, it's green. So if we say that this plant is true breeding for pod color and the color is green, it means that if it is a true breeder, it means that if we allow this pea plant, this plant that we have here, if we allow it to self-pollinate, it's going to produce all the offspring that it produces are also going to be what? Having green pods. Then we say that no, this pea plant here is true breeding for what? For green pod color. Are we together? It's gonna come. We're gonna go into it in more details as we go along. Remember that this topic is 4 weeks, so this is just basically your introduction. Now, walk with me. If this pea plant that we just made, that is white, terminally, what, if this pea plant is true breeding for green pod, it doesn't mean that since it is true breeding for pod color, it is going to be true breeding for what? For other characteristics. No. We look at a specific characteristic. We don't look at the whole organism. I hope that makes sense. If you're having issues with this, please remind me the next time when we're meeting up, when I see your comments, or whenever we start doing Google Classroom. But for now, drop it in the comment section or WhatsApp to me, because I don't want confusion here. I'll give you an example. What example? No, let's leave it like this for now. So, in conclusion, we can only say that a plant is true breeding for a specific characteristic when self-pollination takes place and all the offspring produced have the same characteristic as the plant, as the parent plant. Now, walk with me. After Mendel did all these experiments, some conclusions were brought about from these experiments, and these conclusions are going to be relevant to us as we go on. You need to know these conclusions, right? The very first one was that any hereditary characteristic, in this case, because it was Mendel was doing this, in this case it was plant length in peas, is going to be determined by 2 genes. Let's go back to our thing. Mendel, in his conclusions, realized that no, wait a minute. Any hereditary characteristic, in other words, any characteristic that can be passed on from mommy and daddy to child, from parent to offspring, any characteristic, and in his case he was using peas, remember. He noticed that for any characteristic in peas, stem length, pod color, pod shape, seed color, flower position, flower color, for any of these, it was going to be determined by 2 genes, Pick any characteristic here. Whichever characteristic that you pick, you are going to end up with what? Two genes that control that characteristic. For example, if you look at seed color, you have two genes. One gives you yellow seeds and the other one gives you green seeds. If you look at flower color, you have a gene that will give you purple flowers, and then you have a gene that will give you white flowers. And then if you look at pod shape, you're going to have a gene for inflated pods, and then you're going to have a gene for constricted pods. So this was a conclusion that he brought about, that he came out with when he came to his experiments. That was the very first conclusion that he came up with. Let's keep on moving. Walk with me. The second conclusion that he came up with is that one gene of the gene pair will come from the mother, and the other of the gene pair will come from the father. Remember this. Every time when meiosis occurs, you are going to have what? Haploid number of chromosomes in each gamete. So, if we look at a more complex level, if we look at you as an individual, for each characteristic that you have, one gene came from your mother and the other gene came from your father. And then these make up a gene pair. And it is carried on two chromosomes of a homologous chromosome pair. On two chromosomes of a homologous chromosome pair. Let's stop there. Let's go back to it. Let's just repeat again for emphasis. One gene, Mendel found out that one gene of the gene pair comes from the mother and the other from the father. And the gene pair is carried on on two chromosomes of a homologous chromosome pair. Remember when we did this? Let's go back to meiosis. Revision. Walk with me. Initially, we start with what? A chromosome. And we know that on a chromosome, we're going to have many genes. Is that not so? And then, in a homologous pair of chromosomes, same size, shape, and genetic composition, you are also going to have, so you're going to have in a homologous pair of chromosomes, you are going to have a situation like this. One coming from your dad, and then the other chromosome coming from your mom. And when you are undergoing gametogenesis, these are going to be split. And then this one will go this side into a different cell, and then this one will go to its own side. And then they end up in different cells. So, when fertilization occurs, on each chromosome, in each homologous chromosome pair, you are having two chromosomes on a homologous chromosome pair. You are having two chromosomes, and these two chromosomes are of the same size, shape, and genetic composition. Now, when they have the same size, shape, and genetic composition, and guess what? One of that homologous pair of chromosomes is coming from your father. The other one is coming from your mother. And then during fertilization, these were brought about together. And then you now had a homologous logus pair of chromosomes. What does this mean? And this is only one of many of 23 pairs. So he found that it was Mendel who actually found that, no, wait a minute. For each characteristic that can be inherited, any one of these, there are actually two genes that control that characteristic. So if you look at seed shape, you can either have a gene that will give you round seeds and a gene that will give you wrinkled seeds. That was his second conclusion. Right, let's continue. Thirdly, he found that a particular gene occurs in two or sometimes more different forms that affect the same characteristic in different ways. What are we talking about there? We are saying that the gene that codes for seed color, is found in two forms, yellow and green. The gene that codes for pod color is found in two different forms. Either it's green or it is yellow. Right? Walk with me. These alternative forms of one gene. Remember what the gene is? The gene is either height of pea plant. And the height, you can either have pea plants being tall or short. So in other words, there are two different forms that give you that gene. Now, the alternative forms of the same gene are known as alleles. This is a past exam paper. This is a past exam question. The alternative forms of the same gene are known as alleles. Stop there. Walk with me. I don't want you getting confused. So, here we go. By looking at conclusion number 3, we can see that the gene for stem length or height has two alleles. One allele is for tall and the other allele is for short. Hope that makes sense. Right. Walk with me. So, alternative forms of the same gene are known as alleles, and different alleles contain different information about the same characteristic. Different alleles contain different information about the same characteristic or trait. What do we mean? We are saying that I'm a pea plant. I'm either going to be tall or short, depending on the alleles that I have in my gene pair. They will influence who I become if I'm a pea plant. Right. So this means that in pea plants, the plant length is determined by a gene with two alleles. One allele results in tall plants and the other allele results in short plants. For seed color, another example. This is another example. One allele will produce what? Yellow seeds, and the other allele will produce green seeds. This is what he found, that a particular gene occurs in two or sometimes more different forms that affect the same characteristic in different ways. Let's continue. Number 4. The alleles of a particular gene occur at the same locus on a specific homologous chromosome pair. The alleles of a particular gene occur at the same locus on a specific chromosome pair. I want to stop here because I'm worried about the size of the video now before I upload it to YouTube. But I'm going to continue from number 4. We're going to recap the next lesson and then we continue from there. And then we see what we can do as much as we can with the time that we have. I hope you enjoy these lessons. And please, make sure that you write your notes. Muscle memory. How are you going to know what to write in the exam if you have never written it before? Remember, the aim is for us to excel. So let's stay motivated and let's keep working hard. I'll see you guys. Keep the comments coming and then tell me what's up. If you need anything, you just tell me via WhatsApp on the group. Okay? Peace.